Okay, welcome back. Let's finish up our chapter about embryonic development. So we are to the point of discussing gastrulation. Gastrulation, and we've talked about how it happens in a couple of species. And now let's talk about the steps of gastrulation in a human. So in a human, egg cells are the largest cells that we have, but they are very tiny. They're still very tiny cells, and they have very little yolk. In a human, the blastocyst here is going to be the human equivalent of the blastula. It will be the source of embryonic stem cell lines. The inner cell mass will develop into the embryo, and the trophoblast is the outer epithelial layer of the blastocyst. And it does not contribute to the embryo. The blastocele inside is what's going to become the embryo. The trophoblast on the outside is going to be important in implantation. So at this point, when we're talking about humans, we're talking about an internal development. So this isn't just an egg that's been laid and is going to develop. This egg has to actually implant into the uterus. And so that is the point of the trophoblast. After implantation, the trophoblast is going to continue expanding. You can see it growing, spreading here. It'll form sets of embryonic membranes, and these membranes will enclose the specialized structures outside of the embryo. We'll come back and talk about those embryonic membranes in a minute. We're going to form outward movement of the epiblast through the primitive streak, just like what we just saw in the chick embryos. And after gastrulation, we have our three embryonic germ layers: the ectoderm, the mesoderm, and the endoderm. Okay, so those extraembryonic membranes, these are going to be found in mammals, and these are going to also be found in reptiles, including birds. So we are all called, first of all, these extraembryonic membranes. So these are all important in land vertebrates, organisms that don't lay eggs in the water. Instead, we either are laying eggs on land or developing internally. So land vertebrates have 4 extraembryonic membranes, and they provide a support system for embryonic development. First is the chorion. The chorion is going to function in gas exchange, so oxygen, carbon dioxide gas exchange. The amnion. The amnion encloses the developing embryo in amniotic fluid. When a woman goes into labor, if you've heard of her saying, her water broke, that refers to this membrane breaking and the fluid inside of it flowing out. The yolk sac, this encloses the yolk. This is the yolk sac, which encloses the yolk. And the allantois. And the allantois is going to dispose of waste products and contribute to gas exchange. Okay, so mammals, reptiles, we have this amnion. So we are amniotic organisms. Okay, what structural adaptations in chickens allows them to lay their eggs in arid environments, that's dry environments, rather than in water? A, extraembryonic membranes. B, yolk. C, cleavage. D, gastrulation. Or E, development of the brain from ectoderm. Okay, next let's talk organogenesis. This is the development of organs. So these embryonic germ layers are going to become the basis of all our organisms. The cells from the 2 to 3 layers. Sometimes you could have 2 or 3 layers together working to form one single organ. Sometimes one layer can develop an organ on its own. One interesting thing we'll talk about is neurulation, which is formation of a neural tube. The neural tube is going to produce this brain and the spinal cord. So neurulation begins when the dorsal mesoderm is going to form a notochord. A notochord is one of the 4 defining features of all vertebrates. So this does appear at some stage of vertebral development. The notochord will signal the ectoderm above it to start forming a neural plate. So here's the neural plate. This is called induction. when one group of cells can impact the development of nearby cells. Induction occurs a lot in development. We're not going to spend a lot of time talking about it. But this is how a lot of your body is able to say, okay, here's one body part, then the next part develops, then the next part develops, and the next part develops. So here, the notochord induces the development of the neural plate. As neural plate forms, it will fold inward and form this neural tube. So that's what this middle picture here is, folding inward, and you're getting a neural tube. Then here between the neural tube and the outer layer of ectoderm is a layer of neural crest cells. And these neural crest cells will migrate to form various organs of the body. So it'll migrate to form like the teeth or parts of ectodermal organs throughout the body. Some of these mesodermal cells are also going to be able to migrate from the lateral sides, and they'll form little segmented repeated structures called somites. And those somites have a major role in organizing segmented structures of vertebrate body. In humans, they are involved in forming vertebral discs. A lot of the ability of vertebrates or of animals in general to undergo morphogenesis is because we have cells that are able to move, and entire cells can move, portions of cells can move. You don't see that a lot in plants because of their thick cell wall. But without a cell wall, our cells are a little bit more flexible. And this involves microtubules and microfilaments. If you remember, these are parts of the cytoskeleton. And rearranging the cytoskeleton will change the shape of the cell. I mean, just like if you rearrange the skeleton of your body would change shape. Reorganizing the cytoskeleton forces the cell to change shape. Actin filaments, which is part of your cytoskeleton, are able to contract or kind of tighten up, and that causes them to become wedge-shaped. And this is driving force in the ability of invaginating a cell layer. Okay, what would happen if just before neural tube formation, you treated embryos with a drug that blocks the function of microfilaments? Okay, let's look at a couple of ultrasounds. I know I showed you an ultrasound last chapter, and we had some ultrasounds last semester, but they're too cute to pass on. Okay, see if you can see the baby's spine, shoulders, head. Can you figure out where their ribs are? Here you can see the baby from the side. You can see, they're sucking their thumb. Kind of see the outline of their mouth and their nose. And see, this is their belly area. Okay, and here's an ultrasound at 20 weeks. So at this stage of 20 weeks, this baby is going to look quite human-like, not entirely the same as you and me, but you skipped it. Okay, there's our arm bone, backbone, skull, their mouth, their nose, shoulder, leg. Oh, shoulder again. Here's their leg. Their spine. Their little folded up leg. Okay, and let's watch a video of a frog embryo developing.
Frog development. Let's observe the development of a frog embryo in pond water. Many of the developmental processes seen in a frog are also observed in other vertebrates. In this time-lapse video, you will see the end of gastrulation and the beginning of organ formation, in particular, the beginning of the nervous system, in Xenopus, the African clawed frog, an animal that is a favorite of embryologists. This video picks up the frog embryo late in gastrulation. Cleavage has produced numerous cells too small to be seen individually. The cells roll over the lip of the blastopore and into the interior of the gastrula. Inside, Lighter colored, yolk-laden cells that originated at the vegetal pole form the endoderm of the embryonic digestive tract. Cells from the animal pole spread over the embryo and form ectoderm. In between, a layer of mesoderm is taking shape. This is the back of the embryo with what will become the head at the top. The blastopore later becomes the anus. Under the dorsal surface, from head to tail, a rod of mesoderm called the notochord is forming. Later, it will be replaced by the vertebral column. Above the notochord, the ectoderm thickens, forming a neural plate bounded by two neural folds. This is the beginning of the neurulation, the formation of the nervous system. The neural folds join at the midline, and the middle portion of the neural plate sinks beneath the surface, forming a neural tube. The enlarged anterior portion of the neural tube will become the frog's brain, and the posterior portion will develop into the spinal cord.
Okay, a lot of times in development we see apoptosis occurring. Apoptosis refers to programmed cell death. So to back up and understand, a lot of vertebrates evolved after a body plan had already been established. And rather than scrap the whole body plan and start from fresh, a lot of it was just discard parts that you don't need. So the body will start developing, and we see a very similar plan of development throughout a lot of animals. And then apoptosis allows death to parts of the body that we don't need. So in a human, this would include like webbing between your fingers. We don't need webbing between our fingers. Apoptosis of our post-anal tail. That tail shows up very early in development, but we don't need that. So at various times during development, individual cells or sets of cells or whole tissues will stop developing, will die, and be engulfed by neighboring cells. So one example would be that many more neurons are produced in a developing embryo than will be needed. So the extra neurons are eliminated by apoptosis. Sometimes a structure functions in early stages and is eliminated during later development. This could be seen as a tadpole undergoes apoptosis, losing its tail during its metamorphosis to being a frog without a tail. Okay, one other thing we're going to talk, just a couple of slides more and we'll be done. One thing that's important in animal development is axis formation. So if you're an organism with a bilateral symmetry, you have a front and a back, a left and a right, a top and a bottom. So the dorsal and ventral sides are not symmetrical. Back and belly are very different. Anterior, posterior are not symmetrical. Those are very different. Your head or your tail. The right and left is a line of symmetry. Your right and left are symmetrical to each other. And some of that symmetry starts, and this all starts very early in development. The egg itself already has an animal pole and a vegetal pole established by where the yolk is. Then upon the sperm binding, the cell, like extracellular membranes of the cell, kind of rotate around towards where the sperm bound. And that kind of helps determine where the cleavage is going to start occurring, which will eventually then lead into those axis formation. And then restricting development potential. So in a mammal, if you have an embryo or if you have a fertilized egg that has gotten to the 2 stage or the 4 stage or the 8 stage, you could take out one cell and it could still develop into an entire person. So, if you're at the 4 cell stage and it splits in half, you have 2 cells back at the 2 separate 2-celled organisms. They could turn into two twins. That's how you would end up getting identical twins. But after that 8-cell stage, embryonic cells are not totipotent. They are not able to develop into an entire organism. So if you took a blastocyst at like 100-cell stage and tried to cut it in half and have 2 blastocysts, they would not be able to develop into an entire organism. A lot of their fate has already been restricted and determined. This progressive restriction of developmental potential is a general feature of development in all animals. So by the time you go to being triploblastic or diploblastic, those cells have a specific feature that they are already going to be bound for. And then as it develops more, it is going to develop more and more into this is going to become part of the neural cord. This is going to become part of the spinal cord. This is going to become part of the brain or the heart. And as the organism develops, fate of each cell becomes more fixed. In general, tissue-specific fates of cells are fixed by the late gastrulous stage, so what part of tissue it's going to be. And then as embryonic cells acquire distinct fates, they can influence each other's fates by induction. So one part developing is able to influence how the next part is able to adapt and develop. And the response to an inductive signal is usually to switch on a set of genes, and then those set of genes in that cell are going to be turned on to determine how that cell is going to develop. Your book goes into a lot of discussion. We are barely touching the surface on this. I think this is fascinating, and I would love to talk for hours about how this embryo is going to develop and know that this is its head, and this is its belly, and this is its tail, how each of those develop. And your book does have a nice discussion, and if you'd like to discuss that with me sometime, we can talk about it, or I can send you some references to talk about it, because it's just so exciting. It's just a matter of time for us. But if you do have any questions about, cell fate or anything else in life in general, I am always here available for you. I'd love to talk anytime. I will see you all later. Bye.